Hi, my name is Rob, and today we're going to be going over organic extractions, where we're going to learn the process and some background information as to how they work. First, let's go over some DNA facts. DNA has the ability to enter or remain in a liquid phase. DNA has one negative charge per nucleotide. It has a molecular weight of 1.98 by 10 to the power of 12 grams per mole. There are approximately 3 picograms of genomic DNA in a haploid cell, and there are approximately 6.6 picograms of genomic DNA in a diploid cell. The normal number of leukocytes in human peripheral blood is 5 to 10 million cells per milliliter of blood, which is about 30 to 60 micrograms of DNA per milliliter. The normal number of sperm per milliliter of semen is 1,500,000, which is about 450 micrograms of DNA per milliliter. Additionally, each milliliter of semen can contain approximately 5 million leukocytes, which contribute another 30 micrograms of DNA per milliliter, for a total of approximately 480 micrograms of DNA per milliliter. Let's go over the types of organic extraction. In general, there are two types of organic extractions: stains containing spermatozoa and those which do not contain spermatozoa. The first example that we're going to go over is of an organic extraction of a non-sperm stain. The first step in the process is the solubilization of the stain. In step 1, the stain is being placed into a microcentrifuge tube which contains extraction buffer. This is going to be vortex, centrifuged, and then put to incubate. With the solubilization of a stain, water must be replaced and stains must be resolubilized. It is important to protect the DNA from unnecessary degradation during this process. The process generally involves soaking the stain in buffer, and this is usually done at 56 degrees Celsius for 2 hours. The buffer contains EDTA. EDTA is a chelator of magnesium to prevent the action of the nucleases that could degrade the DNA. Nucleases need magnesium for their activity. Because of the action of EDTA, the magnesium is no longer available, and therefore the nucleases become inactive. The next step is the denaturation and hydrolysis of proteins. The detergent in the stain extraction buffer causes the lysis of cellular membranes and the dissociation and denaturation of the histone proteins that are tightly attached to the DNA strands. Detergents destroy the secondary and tertiary structures of the proteins, and this leads to their decreased solubility in the aqueous solution and increased susceptibility to the hydrolytic activity of proteolytic enzymes. The detergent most commonly used at this step is sodium dodecyl sulfate, which is commonly referred to as SDS. Proteinase K is an enzyme which is also used, and this aids in the hydrolysis of histone proteins. This enzyme is active across a wide range of pH. It is active in the presence of SDS and is unaffected by metal chelators such as EDTA. In the next step of the process, any solid material, which is known as a substrate, is transferred into a spin basket. This undergoes a centrifuge process, and then the basket and substrate are then discarded. During this step, the substrate can be placed into a spin basket to recover the liquid remaining on the substrate. Some laboratories retain the substrate, which can be re-extracted, while others discard the substrate at this step. The liquid is then carried to the next step, which is the removal of denaturation products. In the next step, which is the removal of denaturation products, you combine the extract with equal volumes of phenylchloroform isoamyl alcohol. This is vortexed and centrifuged. Then the aqueous phase, which is the upper level, is removed for purification. The diagram shows the two phases that will be seen at this step in the extraction process. The DNA is found in the aqueous phase because the DNA molecule has a charge. This aqueous layer is then removed and placed into a new microcentrifuge tube. It is important to only remove the aqueous layer and avoid the organic phase and the interface between the two phases. Phenol, which is present in the organic phase, can lead to PCR inhibition. If you do happen to disturb the interface or pipette some of the organic phase, you can simply recombine the phases, centrifuge, and start again. Safety is very important when doing organic extractions. Phenol is highly toxic and should be handled in a fume hood while wearing personal protective equipment. Skin contact and inhalation should be avoided. Chloroform depresses the central nervous system. It is also a suspected teratogen and known carcinogen, and should never be handled outside of a fume hood. Denatured proteins are removed with phenol and chloroform. Phenol is an effective protein denaturant. Chloroform, to a less extent, also aids in this process. The products of the denaturation and proteolysis are soluble in phenol. Generally, most procedures use a phenol-chloroform mixture, which includes isoamyl alcohol. Isoamyl alcohol reduces the tendency of proteins to foam when they're denatured during the shaking process with organic solvents. Some procedures use phenol first, followed by one or more treatments with chloroform to ensure complete removal of phenol. The next step in the extraction process is called purification. In this step, the aqueous phase is placed into a centrifugal filter unit. First, insert a filter unit into a filtrate tube. Add sample and buffer and then centrifuge. Remove the filter unit from the filtrate tube and discard the filtrate. Then, reinsert the filter unit into the filtrate tube again and add more buffer to the sample reservoir, cap, and then centrifuge. Finally, add a small volume of buffer to the sample reservoir to cover the filter with liquid. Invert and transfer the sample reservoir from the filtrate tube to a retentate tube. Centrifuge one more time. At the end, DNA is in the retentate tube. This diagram shows the components of the Microcon system. It is made up of a tube, which could be used as a filtrate or retentate vial, and a sample reservoir. The filter that traps the DNA is contained in the sample reservoir. With the Microcon system, centrifugal force is applied, which causes the sample to pass through the filter. The DNA is collected on the filter, which is later inverted into the retentate vial. The liquid collected in the filtrate vial is discarded. DNA can be recovered from the aqueous phase with an ethanol precipitation or using a centrifugal filter unit. This could be either Microcon or Microcon. Most protocols, however, use the centrifugal filter unit because apart from purification of the DNA, it can also be used to concentrate the DNA. After the samples are purified, the DNA is now ready for quantitation. The other type of organic extraction method is called a differential extraction. Stains containing spermatozoa undergo a differential extraction. Differential extraction methods are used to separate spermatozoa from other cell types. Spermatozoa are more difficult to lyse than other cells, and conditions can be set so that all cells except spermatozoa are lysed. The supernatant containing the DNA from these cells is removed from the sperm cells. The sperm cells can then be lysed separately. In a differential organic extraction, the steps are similar to those for a non-sperm stain, except there are two cell lysis steps instead of one. The first step is the non-sperm cell lysis. Extraction buffer, detergent, and proteinase K are added to the sample and incubated. The supernatant containing the DNA from the lysed cells, which is referred to as fraction 1, is removed after pelleting the spermatozoa. The sperm pellet is often washed numerous times with a buffer to remove excess DNA from this lysis step. If any of the sperm cells are weak or otherwise compromised, these may lyse in the first step, leaving a low level of fraction 2, which is the sperm DNA in fraction 1. The second step is the sperm cell lysis. The pelleted sperm cells are lysed under more stringent conditions using a buffer, detergent, DTT, and a higher concentration of proteinase K, and these are all subsequently incubated. This is referred to as fraction 2. Both fractions are extracted separately with a phenol-chloroform isoamyl alcohol combination and purified in a centrifugal filtration device. Dithiotretol, DTT, reduces disulfides to dithiols, allowing release of the DNA from its protective proteins and further degradation of the proteins by proteinase K. DTT is an essential component for sperm cell lysis because the cell membrane contains a high concentration of disulfides.